Hi, my name is Oppong Afranie. I recently graduated from Ball State University, Muncie, Indiana, United States, with Masters of Science in Physiology and a postgraduate certificate in biotechnology. So at Muncie, I was working with Dr. Sharma, and in his lab, we were investigating the expression levels of APJ and SOX17 in hypoxia gain of function embryonic mouse heart during the development of coronary vessels. This will be my table of content. So, the coronary artery disease, also known as the ischemic heart disease, is one of the most common types of heart disease. It is caused by building up of plaque in the wall of the coronary artery. The plaques narrow down the artery over time, resulting in the partial or complete block of blood flow. The blocking of the blood flow causes shortness of breath, chest pain, irregular heartbeat, and heart attack in the worst case scenario. The coronary artery disease is the third leading cause of death worldwide, and in the United States, about 1 in 20 adults have coronary artery disease. Similarly, it also accounts for approximately 6,000 deaths annually in the United States, which makes this disease the leading cause of death in the country. So, mature coronary arteries have three layers, one being the inner endothelial layer, which forms first, the middle smooth muscle layer, and then the third one being the outer adventitial layer made of fibroblast. During the embryonic development, the coronary vasculature forms in a sequence. First, an immature vascular plexus develops, giving rise to the endothelial layer. Sinus venous derived endothelial progenitor cells migrate over the heart surface and into the subepicardial space. These cells express the APJ receptor and respond to the epicardial ligand, being the labella, which acts as a chemoattractant, guiding their movement into the myocardium as the heart grows. At the same time, the endocardium derived endothelial cells invade in the hypoxic myocardium. Through the VEGF-R2 and the VEGF-A signaling, they undergo splotting angiogenesis to form a coronary capillary plexus. The merging of the SV-derived and the endocardia-derived endothelial population ultimately forms the matured coronary vascular network. So, previous studies indicate to us that the labella APJ and the hypoxia SOX17 are critical signaling mechanisms in the development of coronary vessels. However, it is unclear whether the hypoxia activates the SOX17-mediated coronary angiogenesis from the endocardium. And in this study, we wanted to determine whether the myocardial hypoxia condition upregulates SOX17 and repress APJ signaling to maintain the development balance between the XV-derived and the endocardial-derived coronary angiogenesis. And so, we mimic a hypoxic environment in a molecular level for this study, because hypoxia stimulates the angiogenesis through the HEF1 alpha mediated proangiogenic signaling. So, under normal levels of oxygen or pneumoxia, the HEF1 alpha protein undergoes a polar hydroxylation. The hydroxylated HEF1 alpha protein becomes vulnerable or subjected to ubiquitination by the VHL protein enzyme. The ubiquitinated HEF1 alpha undergoes proteasomal degradation of the VHL. Whereas under low level of oxygen with the hypoxia, the HEF1 alpha becomes stabilized. The HEF1 alpha then translocates to the nucleus and activates its target genes, including the proangiogenic genes, such as the HEF1 alpha. So what all this slide is talking about is, this shows that if we're able to delete the VHL, then there's going to be hypoxic environment being molecularly level mimic in our sample. And for that reason, we aim to establish an a particular mouse colony, transgenic mouse colony, which is the NKS 2.5 Cree VHL flux over flux mouse colony, so that we're able to generate our control and our VHL knockout mice to get the embryos which have a hypoxia gain of function. Also, if we're able to establish our hypoxia gain of function in the embryos, then we will determine if the myocardial hypoxia gain of function stimulates SOX17 and also if it represses the APJ. So, to achieve our aim 1, we set up a breeding strategy to obtain the control and the VHL conditional knockout embryos using the VHL flux over flux and the NKS 2.5 Cree transgenic mice. With that, we then deleted the VHL gene, especially in the cardiomyocyte, because we used the NKS 2.5 promoter. With the VHL flux over flux, our VHL gene was being fluxed at each site, and then we used the Cree, which is the recombinase enzyme, to then delete the VHL gene. Also, to achieve our aim 2 and our aim 3, after setting up the breeding of mouse colony, given us the double transgenic of NKS 2.5 Cree VHL flux over flux, we harvested the embryo from the pregnant mouse and then did genotypes to stratify those that are the VHL knockout groups and the control. We then isolated embryonic hearts from these groups, perform an RNA extractions on them, on the heart, and then perform DNA treatment and a cDNA synthesis. After getting our cDNA, after synthesize our cDNA, we use it for qPCR, and that is going to give us the mRNA levels of HEF1 alpha, the SOX17, and the APJ expressed in the embryonic hearts. Also, after setting up our mouse colony and after harvesting the embryos from the pregnant mouse, we then did genotypes of the embryos to then stratify them as we did previously for obtaining groups and also for obtaining VHL knockout groups. We then had or perform isolation of protein and then perform BCA assay to quantify the protein from these embryonic hearts as harvested. We then perform Western blots for us to quantify the protein levels of F1 alpha, SOX17, and APJ, and also VEGF-R2. And this was our results. So for our results, the F1 alpha, for our results of the F1 alpha level, molecular levels, that's the mRNA levels, it shows that there was significant high levels of control compared with the VHL knockout, which was not supposed to be. The F1 alpha protein levels was significantly elevated in the VHL knockout samples compared to the controls, as the evidence of the Western blot here in this slide shows. This aligns with the known molecular role of the VHL as is the key ubiquitin ligase that targets the F1 alpha for proteasomal degradation under the nomosic condition. So the loss of the VHL, that's knocking out the VHL, then stabilize the F1 alpha protein, allowing it to translocate to the nucleus and drive the hypoxia responsive gene expression. The successful upregulation of the F1 alpha protein confirms the biological effectiveness of the VHL knockout model by mimicking the hypoxia condition. So then we look at the results of the APJ. At the mRNA levels, there was a significant increase in the APJ levels in the control compared with the VHL knockout. In this study, as I said, both the mRNA levels of APJ and the protein levels were markedly reduced in the knockout hearts. This reduction suggests that hypoxia exerts a suppressive effect on the APJ signaling. Since APJ expression is crucial for the sinovenous derived vessel formation, its down regulation implies a likely reduction in the sinus venous contribution to the coronary vasculature in the hypoxic condition. These findings suggest a model where hypoxia favors alternative vascular progenitive pathway. By diminishing the APJ signaling, hypoxia may impede the SV-derived angiogenesis, shifting the development burden to other sources, notably the endocardium. This regulatory stretch could represent a built-in redundancy in the coronary development, ensuring sufficient vasculature even when one pathway is compromised. So, also looking at the results of the SOX17, SOX17 expression was upregulated in the VHL knockout samples, though the change was not statistically significant. The lack of statistically significance may be due to the sampled time, as we harvested the heart in the 16.5 embryonic stage, which may not align with the peak of SOX17 expression, as the system may resist a drastic upregulation due to feedback mechanism, ensuring proper cardiac and vascular development. Despite the F1 alpha being stable, the expression of SOX17 might remain within a certain range to prevent potential development issues. Approximate levels of SOX17 are needed for proper heart development. Despite this limitation, the upward trend is biologically meaningful. It suggests that hypoxia, while repressing the APJ, may potentially induce SOX17 to promote endocardial angiogenesis. Also looking at the results of the VEGF-R2, here shows that VEGF-R2, both findings of the protein levels of the VEGF-R2 and the mRNA support the concept that hypoxia activates a broader angiogenic program. Because from this result, you can see that the VEGF-R2 is being upregulated, which complements the F1 alpha stabilization observed in the VHL knockout heart, forming a coherent narrative that the hypoxia initiates a cascade of pro-angiogenic signals. Hypoxia stimulates coronary growth, expansion, and increase the VEGF-R2 expression in coronary endothelial cells. However, we using the NKS 2.5 Cree model for this study resulted that the cardiomyocyte expresses both the VEGF-R1 and VEGF-R2, but the levels of these receptors are significantly lower than those found in the endothelial cells. Nevertheless, this confirmed that the angiogenic machinery remains active under prolonged hypoxia. So, to conclude on our study, the central hypothesis of this study was that myocardial hypoxia regulates the balance between the sinus venous and the endocardial derived coronary angiogenesis through differential expression of APJ and SOX17, and the results support our hypothesis. The observed suppression of the APJ and the potential upregulation of the SOX17 under prolonged hypoxic conditions suggests a developmental flexibility, highlighting a remarkable adaptability in coronary vessel formation, ensuring myocardial perfusion through redundant pathways. This adaptability may have therapeutic relevance in adult ischemic conditions, where inducing similar compensatory pathways could facilitate vascular regeneration. The findings underscore the importance of temporal and spatial dynamics in angiogenesis. The interaction between the SV and the endocardial progenitors is not static, as it evolves with changing physiological cues. Hypoxia emerges as a key moderator in this interplay, capable of tipping the balance between competing angiogenic routes. So for the future direction, I advise that they have to be conducting longitudinal studies to track the expression of level of SOX17 and APJ from the embryonic stage through adulthood, will provide an insight into their roles during the different developmental phases and in response to different roles of hypoxia condition. Expanding the research to include other signal molecules involved in hypoxia and angiogenesis, such as additional VHF family members. I'd like to acknowledge my supervisor, my PI, my principal investigator, which is Dr. Shamar, and other professors who helped me with this study, and also my other members or colleagues in Shamar's lab, and also to acknowledge Dee, who was in charge of the mouse colony, who helped me very much with the mouse colony, and also with our funding source, which is the NHI R15, which provided this research grant to Dr. Sharma for us to be able to undergo this study. And also, I'm very appreciative to Ball State, who gave me the platform and the opportunity to study in the university. Thank you.